Hello guys, so today our topic is EMP pathway. EMP pathway, Embden-Meyerhof and Parnas pathway or glycolysis. Glycolysis or dissolution of sugar occur in all cells of our body. It occurs in two ways, aerobically or anaerobically. It is only important source of energy for mature RBCs because mature RBCs lack mitochondria, so oxidative phosphorylation cannot occur in these. So they completely dependent on glycolysis for their energy needs. During any strenuous activity, demand of oxygen of our body increases but supply remains low. Then this pathway provides energy anaerobically by producing lactic acid. Glycolytic activity is low in cardiac muscle. That's why during ischemic attacks, prognosis is very difficult. Glycolysis is essential for generation of carbon skeletal of non-essential amino acids and glycerol part of fat. Now let's come to the steps. So glycolysis occur in 10 steps. The very first step is conversion of glucose to glucose 6-phosphate. It occurs by using a ATP and in the presence of enzyme hexokinase or glucokinase. The second step is glucose 6-phosphate to fructose 6-phosphate. It is an isomerization step and occurs in the presence of enzyme isomerase. Then the third step, fructose 6-phosphate to fructose 1,6-bisphosphate. It also utilizes one ATP and occurs in the presence of enzyme phosphofructokinase. It is a very important step. Third step, phosphofructokinase is a very important enzyme. It is rate limiting step of glycolysis and it is the slowest step of the whole glycolysis. The fourth step is cleavage of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are interconvertible as they are isomers, and these steps occur in the presence of enzyme aldolase. Now let's proceed to the fifth step. In fifth step, glyceraldehyde 3-phosphate converts to 1,3-biphosphoglyceric acid, 2 molecules of 1,3-biphosphoglyceric acid in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase by generating 1 molecule of NADH. This NADH further converts to 2.5 ATP during electron transport chain. Now 1,3-bisphosphoglyceric acid further converts to 3-phosphoglycerate in the presence of enzyme biphosphoglycerate kinase by generating 2 ATPs. Let's come to the seventh step where 3-phosphoglycerate converts to 2-phosphoglyceric acid. It is a isomerization in the presence of enzyme mutase. Now 2-phosphoglyceric acids converts to phosphenol pyruvate in the presence of an enzyme enolase. Then phosphenol pyruvate further converts to pyruvate, and here again 2 ATP generates in the presence of enzyme pyruvate kinase. So let's recall substrate level phosphorylation, it means where direct ATP production occurs are step 7 and step 10. Step 7, this step where 1,3-biphosphoglyceric acid converts to 3-phosphoglycerate, and step 10 where phosphenol pyruvate converts to pyruvate. So these are the substrate level phosphorylation. In total, substrate level phosphorylation during glycolysis 4 ATPs are generated. Now except 1, 3 and 10, rest all glycolytic steps are reversible. 1 where glucose converts to glucose 6-phosphate, 3 where fructose 6-phosphate converts to fructose 1,6-bisphosphate in the presence of enzyme phosphofructokinase, and 10 where phosphenol pyruvate converts to pyruvate. And the rate limiting step, as we have discussed earlier, rate limiting step is 3 where fructose 6-phosphate converts to fructose 1,6-bisphosphate. Now let's talk about the energetics. During aerobic condition, when glucose converts to pyruvate, the total energy production is net 7 ATPs are produced.